So we are continuing with the function of the plasma membrane. To just recapitulate, we discussed the main function of the plasma membrane was to transport molecules. And we studied diffusion, which can be simple, that is movement of non-polar compounds down the concentration gradient. It can be facilitated. The molecules are moving down the gradient, but the molecules are using a channel or a carrier to move in and out. Then we studied the movement of ions. The movement of ions through the membrane can take place in two different categories. One is the presence of ionophore. The ionophore mediated ion transport occurs down the concentration gradient. As you can see by the picture, the ionophore is binding with the ion, helping in passing of the ion and thereby liberating the ion from the ionophore in the cytoplasmic side. And then and the other way in which ions can be passed through the membrane is the presence of ion channels. So you can see the ions are moving down the gradient via the ion channels. So in all the three circles I have shown here, you are seeing that no ATP is used. So these all categories are passive transport. When we are coming to active transport usually uses ATP, and the molecules are seen to move against their gradient. So we have a higher concentration of the solute inside and a lower concentration of the solute outside, and the solute is being pumped from a lower to a higher concentration. So it uses ATP. Another form of active transport known as a secondary active transport was that here also the solute is moving from a lower to a higher concentration, but this movement is possible because an ion is moving from a higher to a lower concentration. Here you can see the ATP is not used. So these two categories fall under active transport. The right side picture is again a repetition of what I said, but this is now showing you with examples. So we have the two broad categories of diffusion, passive and active. Sorry, the two broad categories of transport, passive and active. When we are talking about passive, you can see simple diffusion as that happens in case of oxygen. The molecule is moving down its concentration gradient. Now, if you look into the B picture, you can see the presence of an aqueous channel or presence of a channel in the membrane, and this channel is helping for the movement of molecules from a higher concentration to a lower concentration. So this is facilitated. So a channel is facilitating the movement of a particle. Now when we are talking about transporter mediated, so this is simple diffusion through ion channel. This is simple diffusion through transporter mediated. Again, this falls under facilitated as the molecules bind to membrane protein and are transported from one side of the membrane to the other side. So these three, as you can see, non-mediated, here the molecule is moving with the help of a channel, is moving through a channel. Water moves in this way. There are aqueous channels, there are channels in the membrane known as aquaporins. Through these channels, water moves. So in case of A, B, and C, we are seeing the movement of particles from a higher concentration outside to a lower concentration inside. For A, no help is required. For B, a channel is required, so it's being helped. And for C, a transporter is required. And when we're coming to active transport, again, you are seeing that in case of active transport, again, a transporter is required. A protein molecule is playing a role in transporting of the solutes from a higher concentration to a lower concentration with the breakdown of ATP. So you can see that this is the sodium potassium pump, which follows the active primary transport. Now, this is how the basic concept of diffusion is that if there is a membrane and if there is a higher concentration on one side of a solute and a lower concentration of the solute on the other side, the basic philosophy of diffusion is that the solutes will try moving across the membrane unless and until all the solutes have reached an equilibrium. So the concentration of the solutes in this side equals to the concentration of solutes in this side. So this is how the solutes movement, if they're allowed to move freely across a membrane. We have discussed simple diffusion yesterday, and diffusion, as you can see, is the movement of solute from a higher concentration to a lower concentration. The black particles of the solutes are moving from a higher to a lower concentration. And yesterday I clarified osmosis. Some of you also gave me right answers. It is the movement of water. So this is where this particular solution has a lower concentration of a solute and the higher concentration of the solvent. So in osmosis, it is a movement of water from a higher to a lower concentration. So there's a gross difference between diffusion and osmosis. And this picture is showing you an erythrocyte. And in an erythrocyte, via example, we can see how simple diffusion, facilitated diffusion, mediated by channel, facilitated diffusion mediated by carrier, and active transport all are occurring. So let's look into it one by one. The first case is a case of simple diffusion. So by now you've understood what is simple diffusion. So you can see all these gases that are moving across the erythrocyte membrane, they are moving as per simple diffusion. They're moving from a higher concentration to a lower concentration. Here, water molecule has been also shown. Next, just now I mentioned that there are small aqueous channels present on the plasma membrane. These aqueous channels are called aquaporin. They are protein channels present on the plasma membrane, and through these protein channels, they are specific for water. And through these protein channels, water can easily enter the cell. So the rapid inward and outward movement of water is being seen in case of this aquaporin channels. So we saw the case of simple diffusion here. We are seeing the case of facilitated diffusion by a channel protein. Through a channel, water is moving in and out. Now let's look into the another case of facilitated diffusion, that is the movement of glucose across the plasma membrane. You can see that the glucose is moving across the plasma membrane with the help of a transporter which is known as GLUT1. So as this is a part of diffusion, so you can well understand that glucose is moving from a higher concentration to a lower concentration. So this is GLUT1, glucose transporter. There is another anion exchange protein occurring. This also falls under facilitated diffusion. In this case, you can see the inward movement of one molecule, that is a chloride ions are moving in as a result of the bicarbonate ions moving out. So that is an anion exchange protein. So one is moving in, one of the charged molecule is moving in via this protein, whereas another charged molecule is moving out. So the anion is exchanging the chloride ion for the bicarbonate ion. The bicarbonate ion is moving out, the chloride ion is moving in. So that is also an example of facilitated diffusion. And finally, we are coming to active transport. As you can see, it is driven by the hydrolysis of ATP. The sodium potassium pumps, as you can see, is moving 3 sodium ions outward. For every 2 potassium ions, it is moving inward. So the sodium ions are moving outward, as you can see here, and the 2 potassium ions are moving inward. Now, this is a form of active transport. As I constantly said, the ions move against the concentration gradient. This is requiring ATP. Just a second, it got stuck. Now, so we have understood that when we are talking about transport, transport can be passive, active. Passive includes diffusion, which can be simple, facilitated, and active includes transport via pumps. So that was the molecules that moved and how they moved. Now we are coming to another important part, that is these molecules are moving either by their own, as in the case of simple diffusion, or by the help of some channels, some transporters, as in the case of facilitated diffusion, or by the help of pumps, as in the case of active transport. Now, so let us look into the three classical molecules through which the three classical proteins through which the different molecules can move across the membrane. So let us come to the channels first. Channels, now when we are talking about channels, keep it in mind, they permit the movement of specific iron or water down their electrochemical gradient. So that is diffusion. So whatever is moving through a channel is following the philosophy of diffusion. But since it is the molecules are moving from a higher to a lower concentration with the help of a channel, so we can say it is a facilitated diffusion using a channel. And as I said, through channels, only specific ions or water can move down their concentration gradient. So this is a channel, and channels usually have a gate. I'll show you the gates. They usually have a gate. A channel is not constantly an open tunnel. There is a gate. So membrane controls the gate of the channel. Constantly water molecules cannot enter in. Constantly ions cannot enter in. Only when the gate is open, the molecules can move from outside to inside. So the permeability of the membrane is controlled. If the channel was constantly open, then the permeability of the membrane could not have been controlled. Since the channels have a gate which can be opened and closed according to the need of the cell, so that when the need arises, a molecule moves in, and when the need doesn't arise, the channel is shut off. So channels have gates. Now we are coming to the next category of protein molecules present on the membrane which facilitate movement of small molecules or ions. Again, here also we see in most cases the molecules are moving along their concentration gradient, but often it is also seen that molecules can move against their concentration gradient. Now let's look into what happens. When we are talking about transporters, there are two basic categories. The two basic categories is that you can see the first one is called uniporter, and these two together are known as co-transport. So the name of co-transport suggests that two molecules are being transported together. When we are talking about uniporter, you can understand by the picture itself, only one molecule is moving down its concentration gradient. But when we are talking of co-transport, you have to understand that the two molecules are moving in this particular transport system. Just now, while discussing that example of RBC, I showed you the anion exchange protein where the bicarbonate ions are exchanged for the chloride ions. So there were two ions moving in two opposite directions. Now, if I look into the two categories of co-transport available in the plasma membrane, one is known as symport and the other is known as antiport. Sym, that means you can see the red and the black molecule, the red and the black arrow, both of them traveling in the same direction. Both the red and the black molecules are moving from outside to inside. So that is symport. When we talk about antiport, that means the direction of one molecule, the direction of movement of one molecule is in exact opposite to the direction of movement of another molecule. So if you look into the picture, the red molecule is moving in a direction from outside to inside, whereas the black molecule is moving in a direction from inside to outside. It is often seen that if one molecule moves in the direction of higher to lower concentration, so we have the red molecule higher here and the red molecule lower here. So the red molecule is moving from a higher concentrated region to a lower concentrated region. In contrast, we have a higher concentration of the black molecule here and the lower concentration of the black molecule here. The black molecule is moving in the opposite direction. So if the red molecule is moving from a I think so. If the red molecule is moving, it's in the opposite direction against the electrochemical gradient. If let me rephrase this statement once more. If the red molecule is moving from a region of lower concentration to a higher concentration. So we have a higher concentration of the red molecule on the outside. We have no, let's take it this way. We have a higher concentration of the red molecule on the outside. Now, the red molecule is moving along the concentration gradient. So we have a higher concentration of the red molecule on the outside, plus. We have a lower concentration of the red molecule on inside, minus. So the red molecule is moving from a higher to a lower concentration. In case of the black molecule, we are having a lower concentration of the black molecule on the, just a second. We are having, so it's moving against the concentration gradient. So we are having a lower concentration of the black molecule on the inside. So lower concentration of the black molecule on the inside and a higher concentration of the black molecule on the outside. So this is the scenario. So when we are talking about the red molecule, you can see the red molecule is moving from a region of higher concentration to a region of lower concentration. So it is moving down its electrochemical gradient. But when we are thinking of the black molecule, it is moving from a lower concentration to a higher concentration. So it is moving against its electrochemical grade of the electrochemical or concentration gradient. So in most cases, whenever there is an antiporter movement, it is often seen that in co-transport, in symporter cases also, if we think of the same phenomena, it is often seen that one molecule is moving down the concentration gradient and one molecule is moving against the concentration gradient. So this is a sort of secondary active transport. Because we studied in secondary active transport, in active transport, the molecules move against their concentration gradient. Even though the red molecule is moving down its concentration gradient, the black molecule is moving against its concentration gradient. This is for the antiporter. For the symporter also the same thing. So we have a higher concentration of the red molecule here, and we have a higher concentration of the black molecule here. So the red molecule is moving from high to low. The black molecule is moving from low to high. So this is a co-transport. Two molecules are being transported simultaneously through these transporter proteins by the help of these transporter proteins. And finally, we are talking about the third category of molecules or proteins that play an important role in transport of ions or small molecules against their electrochemical gradient. So when they're moving against their electrochemical gradient, they need energy. Here also we saw that same movement, but one movement was buffered by another movement. Here only one ion or a small molecule is moving down its against its electrochemical gradient, and this movement is possible because of the breakdown of ATP to release energy. So pumps use the energy released by ATP hydrolysis. This is ATP hydrolysis that power the movement of ions or molecules against their electrochemical gradient across the plasma membrane. So these are the three basic categories of transport mechanisms involving the three major class of proteins that account for the transport of various solutes across the membrane. This is another simple picture of how showing you, this is a picture from Bruce Alberts, the top one, showing you simple diffusion, channel mediated diffusion, transported mediated diffusion. These both falls under passive transport, and then we have a active transport. Now this is, as I was talking about, that ion channels. Ion channels have gate. They are not constantly open. They are open depending on the need of the cell. So when the this is a gate closed, a single ion channel with a single gate, and when this ion comes on near the vicinity of the gate opens and the ion can enter. Similarly, when we are talking about transporters, transporters also have gate. We just now talked about the transporters. We saw uniport and we saw co-transport that contained the symport, two molecules moving in same direction, or the antiport, one molecule moving in another one direction and the other just in the opposite direction. But transporters also have gates, and they have, as you can see, two gates. One on the outer side facing the exterior and one on the inside facing the interior. And one thing to be kept in mind, both the gates are not simultaneously open. The opening of one gate, for the gate 1 opens, and after a fraction of a second when the gate 1 is closing, then only the gate 2 opens. It is never that gate 1 and gate 2 open simultaneously. There is a time lag between the opening of the two gates as well between the closing of the two gates. So there is always a time lag. They do not open simultaneously. Now, this structure is the structure of an aquaporin. So you can see this is the inside of the cell, this is the outside of the cell. You can see the small water molecules moving through this aquaporin. These are channel proteins, and they facilitate the movement of water molecules across the bilayer. In plants specifically, aquaporins play important role in water transport up the stem and the regulation of transpiration in leaves. So you are aware of the phenomena of transpiration, not getting into that. In animal cells, aquaporin often maintains the water balance in the brain and also helps in excretion of sweat from the skin. One thing to be kept in mind is that all aquaporins they are impermeable to charged ions. They only allow the free, hence the name aquaporin, passage of water without affecting the electrochemical gradient across the plasma membrane. So this was aquaporins. This is a channel protein. Now coming to the sodium potassium pump. If you look into the sodium potassium pump, I've just highlighted the sodium potassium pump because this is one important form of active transport. You can see we are having a higher sodium concentration on the outside. This is important. First, you check the concentration of sodium and potassium, and a low potassium on the inside. Okay. Now, so the pump basically, and we have a high potassium concentration inside and a low sodium concentration outside, inside. Now, you can see by the movement of the pump that it will move sodium out. So we have a higher concentration of sodium outside. So sodium is moving against its concentration, and it will move potassium inside. Again, we have a higher concentration of potassium inside. So it is moving potassium against its concentration gradient. So we are thorough about the movement of the ions against the concentration gradient. Sodium is moving from a lower concentration to a higher, and potassium is also moving from a lower concentrated region to a higher concentration region. Now, when I was talking about pumps, I talked about the transporters, some of the transporters, as well as the pumps have gates. So they have gates, and these gates do not open simultaneously. There's always a time lag between the opening of one gate and the opening of the second gate. Now let's look at the story. So this is the interior of the cell. This is the exterior of the cell. You are seeing 3 sodium molecules binding to the sodium potassium pump on the interior surface, and an ATP molecule has also attached. So once the ATP breakdown has occurred, you can see sodium is gradually, so this gate gradually closes, sodium starts moving outward, and the out gate has opened up. So sodium has moved outward. Meanwhile, this gate has closed. The out gate has opened. Sodium has moved outward. When sodium is moving outward, during that time, simultaneously, potassium binds to the out gate. And as sodium moves out, potassium moves in through the gate. As potassium has moved in, the in gate is still closed, the potassium is moving in. By that time, the out gate has closed, as a result of which the in gate has opened and potassium is released. So there is a conformational change of the in gate and the out gate. So the gate changes its conformation. In one conformation, the in gate is open, in which it can bind with sodium and ATP. The in gate closes, sodium moves out, and the out gate opens. In the out gate open conformation, it can bind to the potassium. Once sodium is released, potassium moves in, the out gate is closed, the in gate opens, and as the in gate opens, it returns to the conformation in which it can again bind to sodium and ATP molecules. So this is how the pumps operate. There's a conformational change in the gates, and as I'm constantly saying, the 2 gates are never open at one time. The opening of the in gate, that is towards the cytoplasm, might occur earlier, and there is a lag after which the out gate opens, that is the gate facing the exterior, and once the out gate opens, the in gate immediately closes. The out gate can only open once only the in gate closes, and vice versa, the in gate only opens after the out gate closes. So this is how primarily molecules move across, primarily how ions are moved from one side of the membrane to the other side of the membrane with the help of pumps. This is another example which I wanted to show you that occurs in cell, which is highlighting the 2 categories of active transport. When we are talking about active transport, we have to remember that the molecules are moving against their concentration gradient. So let us see what are the molecules involved. We have active transport, the one we just now discussed. Movement of sodium and potassium. Okay, so in this case, we have the sodium potassium pump antiporter. Now, in this antiporter, just keep it in mind that we have a high sodium. Sodium is high on the outside and sodium is low on the inside. Okay, and what we are seeing, that sodium is moving outside and potassium is moving inside. We just now saw that. Now, since sodium has moved out and the concentration of sodium is high, to maintain a gradient, the sodium then again travels back through another transporter, which is a symporter. See, we have studied 2 categories of co-transport. I showed you the picture. In one category of co-transport, the antiporter, the 2 molecules move in opposite direction. So potassium is moving inward, sodium is moving outward. So this is the antiporter. In this transporter, you see sodium is moving inward and glucose is also moving inward. So this is a symporter. Now look at the mechanism of the symporter. This is an energy driven process, but this is not an energy driven process. What happens here? You see that Sodium, as I said, has a high concentration outside. Sodium has a low concentration inside. So you can see that sodium is moving from a higher concentration. In this particular symport, sodium molecules are moving from a higher concentration to a lower concentration. That is, sodium is moving down its electrochemical gradient. Once more, sodium is moving from a higher concentration to a lower concentration. So it is moving down its electrochemical gradient. Now, let's look at the scenario of glucose. Outside the cell, we are having glucose. We are having glucose at a lower concentration. Sorry. Outside the cell, we are having glucose at a lower concentration. So, glucose, we have low concentration. Inside the cell, we are having glucose at a higher concentration. So when glucose molecules are traveling, they are traveling from a lower concentrated region to a higher. So this is the scenario of this import. Sodium is moving from high to low. Glucose is moving from low to high. Inside the cell, sodium is low, glucose high. So when the sodium is moving down its electrochemical gradient, this movement is facilitating the travel of glucose against the electrochemical gradient. So you can see sodium and glucose have become couples the sodium. This importer is coupling the transport of sodium and glucose across the membrane into the cytoplasm. So this is how this import is operating, and this is known as secondary active transport. Keep it in mind that in secondary active transport, ATP is not required. But one fundamental principle of active transport will operate there. That is, one molecule will obviously move against its concentration gradient. Here, glucose is moving against its concentration gradient, and this movement is possible because the importer is coupling the movement of sodium also simultaneously along its concentration gradient. So these are the different, this is in a very schematically chart-wise manner. The different categories of diffusion has been maintained, and I'm not getting into details because I've explained them again and again. We have three categories of carrier-mediated transport: the facilitated diffusion, the primary active transport, and the secondary active transport, which can be symport or antiport. Then we didn't discuss this. This is the vesicular transport. We'll come to it. We are gradually entering into that area. In pinocytosis, as I said, small amount of extracellular fluid might be taken in. In receptor-mediated endocytosis, large polar molecules are taken in. In phagocytosis, pathogens are engulfed. And when we are talking about exocytosis, it is the secretory products are thrown out of the cell. I'll show you the pictures. By the pictures, it is easier to understand. So this is what we have discussed till now: the uniport and the coupled transport, symport and antiport. And if you look into the picture on the right, this is a picture to show you that how many categories of transports are constantly occurring within the cell. If I think as a cell as a functional machinery, so you can see there is a simple diffusion occurring there. There are movement of biomolecules in the glycosome. There is movement of biomolecules in the peroxisome. We are having entry of amino acids through carrier proteins. We are having gases diffusing into the biomembrane, through the biomembrane into the cytoplasm. You can see there is this glucose entering the cell via carrier protein. You can see the utilization of ATP out here. So there is also a transport mechanism occurring there. In the nucleus, we can see a lot of biomolecules entering. So see, these are the various transport mechanisms that operate simultaneously within a composite eukaryotic cell. This is a transport mechanism I just wanted to show to show you the fact that glucose, this is the transcellular transport of glucose. Why am I telling the word transcellular? Let us be thorough with the picture. Let us understand the picture first. This orange color is the gut lumen. So it is the intestinal, the gut, the whole of the gut, where our food particles all accumulate. So that is the gut lumen. This is our intestinal epithelial cells. And this is the extracellular fluid. So whenever we take in food, just give me a second. So when we are talking about glucose transport, so this was the epithelial, intestinal epithelial cell. This I just now said. And this side is our extracellular fluid. So when we are taking in food, the food is entering our stomach, moving into the intestine. The food is gradually digested. And we are having the digested product of carbohydrate, that is a broken down product of carbohydrate, or glucose. So glucose is now going to enter the epithelial cell. I just now showed you an example of the glucose sodium symport. And what did I say? Glucose enters from a region of lower concentration. Keep it in mind that the level of glucose in cell is high. So the glucose is entering from a region of lower concentration to a region of higher concentration via the phenomena of secondary active transport utilizing the sodium ion gradient. So we are having a symport here. Glucose is using this symport to move against its concentration gradient. In the cytoplasm, glucose is present. This is the intestinal epithelial cell. Now, glucose again needs to travel to the extracellular fluid because glucose needs to pass to different cells of the body. That is the basic raw material from which you can get ATP. So the moment glucose enters the cell, glucose is in higher concentration here. In the extracellular fluid, the concentration of glucose is low. So here, glucose thus travels across this membrane from a higher to a lower concentration. So glucose is high here, keep it in mind. Glucose is low here, glucose is low here. So initially, glucose moves from low to high via the sodium glucose symport. And now glucose is moving from high to low via the glucose transporter via the transporter that helps in transport of glucose. So here we have secondary active transport. Here we have the passive transport. So this is how glucose is often transported from the gut via the intestinal epithelium to the extracellular fluid. We are seeing on one side. So see, this is the concentration of glucose that they have shown here. This is the apical domain of the intestinal epithelial cells. This side is the apical domain of the intestinal epithelial cells. This side is the basal domain of the intestinal epithelial cells. As you can see, glucose is moving from a lower to a higher concentration via the sodium glucose import, and again the glucose is moving from higher to a lower concentration via the transporter mediating passive transport of glucose. So this is transcellular transport of glucose. And this is showing the sodium glucose symport. As I said, that often these transporters have gates. So the opening of one gate occurs only when the other gate is closed. So we have an out gate facing the extracellular surface. We have an in gate. This is the condition where both the gates are closed and there is nothing here. So this is known as occluded empty. state of the gate. Now both glucose and sodium are being captured. So the gate is open. Now when the gate is open, the out gate is open, both sodium and glucose molecule enter, the gate closes. Because there's a lag, there's a time lag between opening of one gate and the opening of the second gate. This is occluded, occupied. Next, the in gate opens and the molecules come to the inner surface, and again both the gates close. So this is occluded empty. So this is the two state in which the gates may exist. One is the occluded empty, both gates closed, and one is the occluded occupied. Here also both gates closed, but in occluded empty, we do the interior of the transport does not contain the two molecules it is transporting. In occluded occupied, it is containing the two molecules which it is actually transporting. And finally, coming to the last part of the functional part of the plasma membrane, as I was saying, endocytosis. So endocytosis is that some particles are being, you can see the plasma membrane, the particle comes to the plasma membrane. The plasma membrane is pinching inward. So you can see the plasma membrane is pinching inward. As the plasma membrane pinches inward, the particle is gradually entering the plasma membrane, and finally the pinching, this pinching, inward pinching of this plasma membrane then pinches in to form a small vesicle containing the particle. So this is phagocytosis because a larger particle has been taken in. We have engulfment of smaller particles also. See, so these are small particles. They have come in. The plasma membrane is pinching in. It is further moving inward. And finally, the plasma membrane has pinched in to form a vesicle. There are often sometimes receptors which helps in endocytosis. So you can see this is some receptors which have clustered in the plasma membrane. These molecules now bind to these receptors. So this area is pinching in, and finally this buds inward into the cytoplasm. So all these three categories which we discussed just now are endocytosis. Talking about exocytosis, so you can see this is a cellular debris kept in a vesicle. The vesicle starts moving towards the inner surface of the plasma membrane. The vesicle then goes and fuses with the inner surface of the plasma membrane. The vesicle fuses with the plasma membrane, and all the materials inside the plasma membrane are thrown out into the exterior of the cell. So this is exocytosis. And this is a comparison of the transporters and the ion channels. So this is important for you. So what the basic difference between ion channels and transporters? One of the major differences is that transporters are involved in both passive and active transport. Ion channels are involved in passive transport. This is another important thing. The transporters on binding to the solute undergo conformational changes, just as we saw in case of sodium ion, where we saw a change in the sodium binds to the in gate and ATP also binds to the in gate. Then sodium runs in through the transporter, the out gate opens, so there's a conformational change. But sodium is liberated, potassium can then only bind, potassium moves in, the out gate closes, then only the in gate opens. So there's a conformational change in the in gate and the out gate depending on only when at a particular conformation sodium can bind. Sodium cannot constantly bind to the in gate. Only when the in gate reaches a particular conformation, it is possible for sodium to bind inwardly. Same thing, only when the out gate reaches a particular conformation, it is possible for the potassium ions to bind to the out gate. So conformation of the in gate and the out gate is very important. These are some properties of the ion channels for you all to go through. So we'll start with cell junctions. The next day we'll stop our class here.